Hello everyone, welcome back to my channel. So in this video we will discuss about the general aspects of amino acid metabolism. What is the structure of the amino acid? We have NH2, carbon, COH, a variable R group and hydrogen. Fine. During the amino acid metabolism, the amino group is converted into urea and the part which is left behind is called as the carbon skeleton. So, how can you define urea? Urea is defined as the end product of protein metabolism. So, how the urea is defined? Urea is defined as the end product of protein metabolism. End product of protein metabolism. This urea is then excreted from the body. Now the question arises, what happened to this carbon skeleton? This carbon skeleton gets converted into the keto acid. The keto acid which is formed has the various functions to perform. One function is the production of energy. Next is the synthesis of glucose or fat. It is also used for the synthesis of non-essential amino acids. Synthesis of non-essential amino acids. Non-essential amino acids are those amino acids which are synthesized in the body. Whenever we talk about the metabolism of amino acid, we consider the two processes with us. One is transamination and the other one is deamination. In this video, we will only discuss about the process called as transamination. So let's get started with it. We will start with transamination. Fine. Transamination refers to the shifting of the amino group. This is the amino group. So the transamination will refer to the shifting of the amino group from the pair of amino acid to the pair of keto acid. Fine. This transfer or shifting of the amino group from the pair of amino acid to the pair of keto acid is catalyzed by the enzyme called as amino transferase or transaminase. Fine. The transamination is the initiating process in the protein metabolism. The transamination is the initiating process in protein metabolism, means it is the first step in protein metabolism. What is the first step in protein metabolism? Transamination. What does transamination refers to? Transamination refers to the transfer of the amino group from the pair of amino acid to the pair of keto acid. Fine. Then the transfer of the amino group is catalyzed by the amino transferase or transaminase enzyme. These enzymes require the coenzyme called as PLP. We will talk about it in detail. What is PLP? PLP is a coenzyme. which is derived from vitamin B6. So what is the full form of PLP? It holds the full form as pyridoxal phosphate. Pyridoxal phosphate. It is a coenzyme which is derived from vitamin B6 and is required by the enzyme transaminase. Fine. That now we will discuss a few features of transamination. Features of transamination. Basically, we have to discuss few points about transamination. The first point we have already discussed, that the enzyme transaminase we require PLP, which is pyridoxal phosphate, which is a coenzyme and it is derived from vitamin B6. Next point is that transaminase, that is a process called as transamination. I can write it with the TR. The process transamination is very important for the redistribution of amino group, plus it is very important for the production of non-essential amino acid. As the non-essential amino acid will form from the keto acids. Fine. Next part is that the transamination process will divert the amino acid towards the energy formation. How can we conclude that? Look, the transamination process will lead to the formation of keto acid, and we have already discussed the function of keto acids. The function of keto acid also includes the production of energy. That's why it is stated that the process which is known as transamination diverts the amino acid towards the energy formation. Next, in transamination, there is only the transfer of amino group. There is only a transfer of amino group and there is no liberation of free ammonia. This point is very important. What we have in transamination there is only a transfer of the amino group and there is no liberation of ammonia. Fine. Then we have learned that the enzymes are specific in their action, hence the specific transaminase enzyme, the specific transaminase enzyme are there for the specific pair of amino acid and keto acid, which means there are various transaminase enzyme available for a different pairs, but we have two transaminase enzymes which are very important in biochemistry. The first enzyme is alanine transaminase and the next enzyme is aspartate transaminase. These two transaminase enzymes are very important. All the amino acids will undergo the process of transamination except the few amino acids. So proline, hydroxylproline, lysine and threonine. I repeat, proline, hydroxylproline, lysine and threonine are the amino acids which do not undergo the process of transamination. So, which enzymes do not undergo the process of transamination? These are proline, hydroxylproline, lysine, and threonine. Now, basically, the amino acid will undergo the process of transamination to concentrate the nitrogen in glutamate. Glutamate is also the amino acid. Glutamate is the only amino acid that will undergo oxidative deamination. To liberate free ammonia. I think this point is not very much clear to you. This will get clear when we discuss about the process of deamination. So basically, you just have to know that glutamate is the only amino acid that will undergo oxidative deamination in order to liberate free ammonia for the urea synthesis. Fine. There is one noteworthy point. The process of transamination is not restricted to alpha amino acid. Is not restricted to alpha amino acid. We require an example to prove it. We have the del amino group of ornithine. Del amino group of ornithine. This will show up the process of transamination. How del, like one carbon COH, this will be alpha, beta, gamma, del. Here NH2 is connected. So we have this, the carbon that is connected to COH will be the alpha carbon, then beta, then gamma, and then del. Fine. So the del amino group of the ornithine will undergo the process of transamination. Fine. Transamination is a reversible process. Transamination is a reversible process. Fine. The enzyme serum transaminase is very important for diagnostic purposes. Serum transaminase. These are few noteworthy points. Fine. You just have to learn them. Then we will discuss about the mechanism of transamination. We will discuss about the mechanism of transamination. The transamination process will take place in the two steps. Fine. The number one is the attachment of the amino group with the PLP, that is pyridoxal phosphate, and to convert it into pyridoxamine phosphate. What is the first step? First step is the attachment of the amino group with pyridoxal phosphate to form pyridoxamine phosphate. Fine. This is the number one step. Next step is the transfer of the amino group from the pyridoxalamine phosphate to the keto acid in order to form the new amino group. Or you can write that there is a transfer of amino group of pyridoxal phosphate to the keto acid to form new amino acid. Fine. So, what are the two steps which are involved in the mechanism of transamination? The number one step is the attachment of the amino group with the PLP, that is pyridoxal phosphate, in order to form pyridoxal amine phosphate. Next is the transfer of the amino group of the PLP to the keto acid in order to form the new amino acid. Next, we will discuss about the formation of skiff base linkage. The formation of skiff base linkage, and we will discuss about the ping pong biomechanism. Fine. So we know that the enzyme transaminase require a coenzyme PLP. Fine. The aldehyde group of pyridoxal phosphate will bind to the epsilon amino group of lysine residue, which is located on the active site of transaminase. The epsilon is the position of the carbon. So epsilon is that carbon to which NH2 group is connected. The aldehyde group of the PLP, that is pyridoxal phosphate, get connected to the epsilon amino group of the lysine residue, which is located at the active site of transaminase, in order to form a skiff linkage. Which linkage? Skiff linkage. Fine. So when the amino acid comes into the play, what it will form? It will form amino acid, PLP, and the skiff base linkage is there. This will present at the enzyme with the help of non-covalent bond. And this will get tightly bound to the enzyme. Fine. What is the ping pong biomechanism? Ping pong mechanism says that there is the formation of various intermediates. There is a formation of various intermediates in the transamination reaction. Fine. The ping pong biomechanism is suggested by Snell and Brostein. Fine. Now we will discuss about the clinical significance of the transaminase enzyme. The two enzymes we have read. The number one is alanine transaminase. Alanine transaminase, also written as ALT, it is elevated when there is a problem with the liver. So alanine aspartate is elevated when there is a liver problem. Fine. Then the other enzyme, the other transaminase which we have read is aspartate transaminase. Aspartate transaminase is elevated when there is a cardiac disorder. when there is a cardiac problem or there is a problem related to the heart, it is elevated in that case. Fine. So this is all for this video. Thank you for watching.